Welcome back. Now we had discussed about zygotine till now. Now we are going to move to the stage pachytene. Now as synapses and synaptonemal complex were the characteristic words which were there in zygotine, what would be there in pachytene? It would be crossing over. Now if you remember, I used two words, one was crossing over and other was recombination when I talked about the variations which come in sexually reproducing organisms. So basically this crossing over is going to take place in pachytene stage. Another thing to be remembered in pachytene is recombinase. Please pay attention that ase is coming in the end, that means it must be an enzyme. Whenever there is ase in the end, we learn it that it is an enzyme. And what is written before it? It is recombinase. That means the enzyme which would be responsible for recombination. And how does recombination take place? It takes place with the help of crossing over. That is the process which is responsible for bringing recombination. Now pay attention that it was joined over here like this. This is in depth we are seeing that how this chromatin material is attached. Coming back to the cell itself, supposedly there are many homologous pairs. Now see, this is one chromosome. Non-sister chromatids are interacting at synaptonemal complexes. At various synaptonemal complexes, what would happen is that recombinase would come along with other enzymes which cut the chromatin material and cause its crossing over. Now what is crossing over? Supposedly you had a synaptonemal complex over here. Now recombinase would come and take this part of sister chromatid to other one and this would shift to the other side. Now you see the chromosomal composition has changed somehow. The green part has gone to the red sister chromatid and the red one has come to the green one. Now this particular thing is known as crossing over. Alright. Now this is with the help of recombinase. Now there are many places where we did this, these synaptonemal complexes would be there. So there would be crossing over which would be taking place. And this takes place specifically in the case of telomeres. Now what are telomeres? These are the ends of the chromatids, okay? Sister chromatids, they have their endings, they are known as telomeres. So towards the telomeres, what happens? The sister chromatids get interchanged. Red one has gone to the green one and green has gone to red one. This is what is known as crossing over, but yet they are still joined to each other. So what you need to remember in pachytene is that this is the stage where actually the crossing over or the transfer of genetic material takes place between two sister chromatids, not anywhere else. It's between the two sister chromatids within the cell only. Now this has an important role to play in bringing about variation. We'll see how. In the end you'll get an idea of it. So it must have taken place over here as there were many synaptonemal complexes present. So at many places recombinase goes and brings about crossing over. When we shift to the next stage, that is diplotin, what happens is that crossing over has taken place, but the point of junction where the crossing over takes place, they remain intact. That means the homologous chromosomes which have undergone crossing over, they would remain joined at the points like this. There would be a joining. This is joined to this and this one. This particular cross-like structure, it appears like this. This green is going over here and this red is coming over here. So this cross-like X-like structure, which we are looking over here, this is known as Kiasmeter. Alright, so when I talk about diplotin, you have to remember Kiasmeter as the characteristic term of this particular stage of prophase 1. Alright, what are Kiasmeter? These are X-like structures in which the homologous chromosome chromatids, non-sister chromatids, they remain attached at the point of crossing over. So that is known as diplotin. Now there is another point that you need to remember, that in many cells, the cells stay at diplotin stage only until and unless they get mitogens which could bring about terminalization of this X-like structure. So when the terminalization of this X-like structure takes place, that is known as Diakhinesis. So what is to be remembered? Terminalization means ending. That means this X structure, if they are together, let them go separate. Let us have a complete sister chromatid which we had after crossing over. Now there would be no point of contact between the two. Both the sister chromatids are going to be separate, and now they can separate forever if they want to. This is what I get as a result of crossing over. Now we started in the beginning with a uniform sister chromatid. Now it has undergone a little bit of change, and that is seen during the stage of diakinosis. Please pay attention that a few terms are to be remembered, that many of the oocytes, they are arrested in the diplotin stage. That means terminalization of chiasmata does not take place in this case. Then in diakinosis, whichever cell enters the diakinosis, it would undergo the further divisions, that is metaphase, anaphase and telophase would take place inside that cell. It might get temporarily arrested in diplotin stage. Like female oocytes, they are arrested in diplotin stage for almost 10 to 13 years till the females attain the age of puberty. Alright, then we have zygotine. There is another term which is to be remembered, that is very important, that is formation of a bivalent. Now I told you in the zygotine that basically this thing happens, the homologous chromosomes, they come together. So this is an arrangement known as bivalent, where you have 2 centromeres and 4 sister chromatids brought together. That means 2 homologous chromosomes are in close proximity to each other, and that is known as a bivalent. In terms of sister chromatids or the chromatids which are present, it is known as a tetrad. So all in all, this particular stage of cell division, that is prophase 1, is quite complex, and it has resulted in changing of the genetic material which is present, of course that is DNA, inside the chromosome. There is a little bit change, that is, as you can see, as a result of crossing over. This is the prophase 1. Now we are going to see what happens in metaphase 1. Over here we have a totally different scenario as was seen in mitosis, because we have taken into account homologous chromosomes. Now as the tetrads are being formed or bivalents are being formed, how would be the arrangement of these tetrads when we see a cell in metaphase stage? The arrangement would again be of homologous pair of chromosomes. Now instead of a single chromosome with its 2 sister chromatids, a bivalent would come in the equatorial plate. This is one, and the other one would be its homologue. Alright, so the pairs would be arranged in such a manner, and centrioles would have arranged themselves at the poles. Because it is metaphase, the spindle machinery would have assembled and it would come and find the kinetochores. Now pay attention over here as well. Spindle machinery would come in the same way, but it would be in search of kinetochores. Such is the arrangement that spindles coming from this side would come in contact of this kinetochore, because this one is not available for them. Just for learning purpose, we don't question their availability or other things, but if the spindle is coming like this, you can very well imagine it would come and join over here, and it would come and join over this kinetochore. So the 2 different kinetochores of 2 different chromosomes would be attaching the spindle fiber to themselves. Equatorial plate is being formed in the metaphase. Spindle fibers come and attach, and when the transition takes place to anaphase, these spindles, they retract and bring one of the homologous pair of chromosomes, brings one of them towards the opposite pole. So when we see anaphase, instead of centromere getting split and the sister chromatids getting separated, what is being separated? Entire chromosome with its replicated content has move towards the poles. Now you can very well imagine that if we talk about the DNA content as we talked about it in terms of grams, so what we have is that chromosome number is going to be half after cytokinesis, but the DNA content would be similar, but the replicated one would be half of it. So what we have during anaphase is that we have 3 chromosomes which have gone to one pole and 3 which have gone to the other pole. Now you can make it out that during anaphase the chromosome reduction has taken place. We started with 6 chromosomes in the beginning, 3 homologous pairs. Now one of each pair has gone to each end. When the telophase gets completed, what you have is 2 nuclei which are present inside the cell with the genetic material. Alright, this 2 nuclei and the decondensation of that chromatin material has also taken place. So we have 2 nuclei. This would be followed by a simple process of cytokinesis as we had discussed earlier. Now what we have is that supposedly there was 4 grams of DNA in the beginning of the cell division, that is in the prophase 1. Supposedly there was 4 grams of DNA. It needs, let's take it to be 8 grams because 4 grams it must be in the beginning of interphase. After S phase, the DNA content doubled, it became 8 grams. Now what has happened, because the half of the chromosomes have gone to one nucleus and other have gone to other, so the chromosome content would have, genetic content I would say, would have again reduced to this much, half of it. But there is one problematic thing that comes into being is that this, out of these 4 grams, 2 grams and 2 grams are copies only because you have not separated the sister chromatids over here. What is the point of keeping this DNA with its copy in the daughter cells? So now the next division that is going to take place will divide these sister chromatids as well, so that each individual cell gets half the genetic content with half the number of chromosomes. So in the first division of meiosis, that is meiosis 1, what we have is we have half the number of chromosomes going to each nuclei. When the meiosis 1 is completed, you're going to get 2 daughter cells with half the number of chromosomes. Supposedly it started with 6 chromosomes. Now both the daughter cells are going to have 3 chromosomes. But if we talk about genetic content, we had discussed in the earlier class that in meiosis what we do is that the genetic content is also reduced to half and the chromosome number is also reduced to half. So how are we supposed to reduce the genetic content as well? This much, these chromosomes are yet having 1.5 supposedly grams, if 1.5, that is to, let's consider 3 chromosomes, so there would be 6 sister chromatids. There are 3 chromosomes, we took it, there would be 6 sister chromatids. Out of the 6, 3 are the original one, 3 are the synthesized ones. So that is an exact carbon copy of that. So we need to separate those carbon copies as well. Now each of these cells individually would enter meiosis 2. Meiosis 2 is as simple as meiosis 1. Again, there would be 4 stages, same way, prophase 2, metaphase 2, anaphase and telophase. Now, each of these cells individually would enter the meiosis 2. Now in meiosis 2, 4 stages have to take place, and what has not happened in meiosis 1 that has to take place in meiosis 2, that is centromere splitting. Centromere splitting could not take place during the anaphase of meiosis 1, that has to take place during this particular division, where what we are going to see is, we are also going to consider now one of the cells I am going to take. Supposedly this is the cell, alright, it is having 3 chromosomes after first division. Consider it, I am not going to draw it like this, let it be a simple condition like this, which is having a crossed over chromatid over here, alright, Then the other green would be here. Consider it also being crossing over has taken place, and then there is another chromosome. Here also we see crossing over. And the other cell, how it would be? Other cell would also be having crossed over chromosome. We had taken red color, so again I'm going to take the example. This is one homologous, so it is having green part, other one being like this. So this part would be green because I'm trying to show crossing over with this color choice that I'm making and drawing the sister chromatids. This is just to make you understand that the crossing over has taken place. That means chromosomal content has somehow been changed due to prophase 1. Now this cell would undergo meiosis 2, each of them. What would happen? First of all, there would be chromosomal condensation during the prophase 2. Same way, all the chromatin material which was decondensed and it became tangled again into a very condensed form, as we call it, decondensed form, not the condensed form. It would again start condensing and form the chromosomal material. Now such would be the case in metaphase, that these chromosomes are going to come in the center and they are going to form the metaphasic plate where all the chromosomes are present in the center of the cell. Similar way, this centriole would develop spindle fibers. I'm not elaborating it much because you know in what happens in mitosis, same thing would happen over here, that the spindle fibers would take all the chromosome towards the end, and in the end we are going to get two cells with a single sister chromatid. Single chromatid would be present. We are going to get two cells having 3 each. Now here I am considering this thing, that one would be this, I am taking this one, sorry, 1, alright, other is also green, and this. One cell would have half of it. One sister chromatid has gone over here, and in the other cell, that don't consider this one, we are talking about this one only, and rub it for understanding purpose. This cell has further undergone next steps of division, where what we have is the sister chromatids have also separated as a result of centromere splitting, and over here we are going to see the crossing over, that is recombination is also quite visible. So we have chromosomes or the sister chromatids like this. So what we get in the end is 4 different cells with an altered genetic material which show crossing over with half the number of chromosomes and half the genetic material which was in the beginning of the cell. The cell started with the supposedly X amount of genetic material. Now each cell is going to have X by 2. That means the chromosome number has also reduced to half. At the same time, the genetic material has also been reduced to half. This is all about meiosis. Now what is the significance of meiosis? You need to understand it that the ploidy level can be restored only by the process of meiosis. Otherwise, sexual reproduction would lead to alteration of the ploidy of each organism. So these two divisions, mitosis and meiosis, are very important. They are very vital for the cell reproduction. We had these questions, we had all these questions answered, and at the same time we also learned what meiosis is all about, how does it take place, and what are the consequences of this meiosis, what are the various stages of prophase, that is also very important, and what are the characteristic features of